सर प्लांट फिजियो के लेसन्स का रिवीजन एक बार ले लो तो।
पूरा करना मुश्किल होगा बेटा, 3 चैप्टर्स हैं एंड आई विल रिक्वायर मोर देन 6 चैप्टर्स।
फोटोसिंथेसिस ले लें, फोटोसिंथेसिस।
फोटोसिंथेसिस उसमें देखिए इंट्रोडक्शन में साइंटिस्ट के बारे में है, ठीक है, कंट्रीब्यूशन ऑफ साइंटिस्ट, राइट। देन फॉलोड बाय दिस यू टॉक अबाउट लाइट रिएक्शन, then followed by that you have dark reaction, then you have Hatch and Slack pathway, that is C4 cycle, and then you have C2 cycle, and finally factors affecting. कौन सा टॉपिक चाहिए इसमें बोल के? Which topic in this? Or you want me to do everything from this chapter? But एक ही chapter हो पाएगा if we take entire chapter. Which is exclusive difficult topic in this? You tell me. I'll explain that one. Immediately we'll start. जल्दी जल्दी बोली लगाओ, बोली लगाओ.
लाइट रिएक्शन।
Light reaction. Okay. Okay. This was actually proposed by Mr. Robert Hill. सबसे पहला नीट क्वेश्चन बनता है, who has discovered it. ठीक है. Since Robert Hill discovered it, इसको हम लोग बुलाते हैं Hill reaction. It's also known as light dependent reaction or light reaction directly बुलाओ, या फिर you call it as Z scheme. ठीक है. या फिर you call it as non-cyclic photo phosphorylation. The basic idea over here is, see, when we listen to the definition of photosynthesis, क्या है definition of photosynthesis? Can anyone tell me? Definition of photosynthesis? स्कूल वाला definition ही बोल दीजिए आप लोग. Fast, fast, fast. टाइम हो रहा है, क्लॉक इज क्लिकिंग. अच्छा मैं ही बोल दूं. Listen, school wala definition is phenomena where we prepare food. This is what your school teachers have taught you, right? But actually, it is a phenomena where light energy is converted into chemical energy and then we prepare food. Two dialogues are there. Light energy getting converted into chemical energy. This is the first part of it. Second part is then we prepare food out of it. ये जो first part है नी, light energy. What is light? Basically, it is present in form of photons. Okay? These photons, photonic energy, is converted into chemical energy. What is chemical energy? ATP, adenosine triphosphate. The word phosphorylation over here is referring for formation of ATP. So literally, we are talking about how exactly ATP is being generated in the so-called plants. Where and how? Let us try to explore it. See, guys, this is a special cell. See, this is epidermis of a plant cell. सबसे ऊपर वाला layer. I'm taking cross section of a leaf, and then I have enlarged the cells in microscope. The upper part I see that you have palisade parenchyma. Elongated cells are there, called as palisade parenchyma. Right. Then you have spongy parenchyma on the lower side. Both these parenchyma cells, inside them we have chloroplast. Let me enlarge chloroplast over here. See, this is chloroplast, which is double membranous structure. Inside chloroplast, I actually have coin-like structures. ये जो coin-like structures हैं, इनको हम लोग बुलाते हैं thylakoid. Let me enlarge this thylakoid. एकदम deep microscope में ले जा रहा है आपको. अंदर, if I see thylakoid again, it is a double membranous structure. Okay, this double membranous structure literally has got central cavity called as lumen, and inside lumen you have South India's famous dish idli. Right? यहाँ पर idli जैसे structures हैं. Okay, you have these idlis. Don't call them as idlis. As I said, it is known as quantasomes. Quantasomes. Quantum physics, something related to light. Quantasomes, something who's gonna trap light. यहाँ पे I'll take out one quantasome out and then enlarge it. Inside quantasome I have pigments. कौन से वाले pigment? Chlorophyll A, chlorophyll B, xanthophyll, and carotenoids. Carotene basically. Okay? So these four pigments are actually there in the plant who is gonna trap the light and prepare what? ATP out of it. So this is the intro of this particular part. Without understanding this, moving ahead would have been like little bit difficult. Now, let me tell you, I told Hill reaction क्यों बुलाते हैं? Because Robert Hill is the one who proposed it. Why is it known as light reaction? Because it is dependent on light. Light के ऊपर depend होंगे. तेरे बिना जिया जाए ना. So they literally depend on light. Light के बिना जिया जाए ना. Z scheme क्यों बुलाते हैं? This cycle is actually occurring in the thylakoid membrane. It is important need point. कहाँ पे occur हो रहा है light reaction? In the thylakoid membrane. In the membrane, if I see, it literally looks like a Z. That is why it is known as Z scheme. Why non-cyclic photophosphorylation? Photophosphorylation light के ऊपर depend होगी ATP हम लोग बना रहे हैं. Non-cyclic because electrons which are released will never come back. Let me explore this cycle. ठीक है? See, light radiation is coming from the sun. दूसरे चैप्टर से मैं कनेक्ट कर रहा हूँ आपको. In your syllabus, you have learned about photosynthetically active radiation. What is this photosynthetically active radiation? See, this is our Earth. Earth is covered by ozone. The radiation which are coming from sun, less than 50% are filtered and then they come to the Earth surface. कितना radiation Earth को आ रहा है? Less than 50%. And when you talk about trees on the Earth, less than 10% of light is falling on them. And when you talk about so-called these pigments who are present inside quantasome, less than 2% of light is falling onto them. ये जो less than 2% light है ना, this is known as PAR, photosynthetically active radiation. So the pigments which are there inside quantosomes, they are aligned in particular fashion. इसको हम लोग बुलाते हैं light harvesting complex, or I can simply call them as pigments. Okay, light harvesting complex, and you have a leader for this light harvesting complex. जैसा who is your favorite player in Indian cricket team? बोले तो या तो will listen the name of MS Dhoni or will listen the name of Virat Kohli. तो यहाँ पे जो pigments हैं, out of this, who is the captain? That is chlorophyll A. ये जो है विराट कोहली है of the team. Okay. So what's gonna happen? Light radiation, photosynthetically active radiation, light. This marker is light. Okay, it's gonna fall on the pigment. Because of which the light energy is received by them and they start vibrating. Okay. You might have seen school dancing, जहाँ पर what do you say, a group is dancing and one person starts dancing and suddenly he'll stop and show the fingers to the next person and he starts dancing. He'll show to the third person. In this way, the pigments literally will transfer the so-called energy finally to the hero of the team. That is chlorophyll A. Once chlorophyll A receives this energy, the electrons which are there in chlorophyll A will excite from ground state to excited state. इसके electron excite हुए हैं chlorophyll A के, and chlorophyll A is reaction center. As soon as electron become excited, यहाँ पर pigments जो है excite हो गए. यहाँ पर I have photosystem 2. इधर पे है photosystem 1, the proteins which are there in the membrane. And here I have a special complex called as photolysis of water complex. First neat question over here with respect to this is, what is the position of photolysis of water complex? क्या है position? It is towards inner wall of inner membrane. Please note it. Photolysis of water complex कहाँ पर है? In the thylakoid membrane. Where in thylakoid membrane? Towards inner wall of inner membrane. So the light radiation which was absorbed by chlorophyll A will become excited. The electronic energy literally is touching the photosystem 1. It's like you go to temple. You all have seen a temple. नहीं? There is a bell hanging over there. जैसे छोटे बच्चे, they see the what is a bell, they feel like jumping and hitting it. यहाँ पर यही हो रहा है. When the chlorophyll A will excite its electron, it's literally jumping and hitting the so-called base of photosystem 2. Once they hit it, the energy of light is transferred to this complex, which is known as photolysis of water complex. Guys, term is telling you photo means what? Light. Lysis बोले तो तोड़ना, breaking. Light use करके you are breaking what? Water. When I break water, water is H2O, right? So there are two protons and two electrons, and half oxygen is liberated. In intro of this chapter, एक आदमी का नाम आता है who actually told that the so-called water is donor of oxygen. His name is Mr. Von Neal. It is the last scientist in the contribution, okay, जिसने purple sulfur bacteria और green sulfur bacteria पर काम किया है. He is the one who told water will donate oxygen in the plants. Carbon dioxide will make glucose. It is his contribution. आ सकता है नीट में क्वेश्चन, ठीक है. तो यहाँ पर हम लोग बता रहे हैं लिटरली water is donating what? Oxygen. Guys, protons, electrons and oxygen is being liberated. The electrons will now from photosystem 2 will jump to excited state. किसके electrons आ रहे हैं? Not of chlorophyll A, it is of water. So 2 electrons of water are literally taken up by pheophytin, a protein present in the membrane. यहाँ पर जो है, we have a protein called as pheophytin. Pheophytin is gonna pass electron to plastoquinone. Name of the protein is plastoquinone. Plastoquinone, since it is towards outer membrane, यहाँ पर है, it is literally here towards outer membrane. And outer membrane के पास there are huge protons. Even inside the so-called lumen we have protons. Even outside we have protons. तो ये जब negative charge लेता है, who? Plastoquinone. Once 2 electrons are transferred to plastoquinone, it becomes negatively charged. अगर negatively charged है, तो it is gonna attract 2 protons. ठीक है, 2 electrons will pull 2 protons. Positive, opposite charges will attract बेटा. तो यहाँ पर protons are being pulled. Once they take proton, it is only for some time that they will retain it. And now they are transferring the so-called electrons to cytochrome. And what's gonna happen over here? Cytochrome जैसे ये proton लेता है, it will pump it inside. When cytochrome is pumping huge amount of protons, यहाँ पर generate होता है PMF, proton motive force. Guys, I have a question over here. Protons की अगर बात करो, what do you think? Protons is property of acids or it is property of alkali? बेस की प्रॉपर्टी है या एसिड की प्रॉपर्टी है प्रोटॉन्स का रहना? गाइस प्लीज इंटरैक्ट, नहीं तो मैं सो जाऊंगा यहाँ पे.
Acids.
Acid, exactly. The proof of this is H2SO4, HNO3, HCl. When you talk about acids here, only protons are more in it. वही अगर आप बेस की बात करो, OH मॉलिक्यूल आता है ना, NaOH, KOH. Here we have added too many protons. मतलब ल्यूमेन में एसिडिटी बढ़ा दिया हम लोग. आपको भी एसिडिटी ज्यादा होगी तो क्या करोगे? पहले घर में दिवाली सेलिब्रेट करेंगे ना, पटाखे बजाएंगे. तो सिमिलरली यहाँ पर भी too much of acidity has happened, so they have to release it out. तो कहां से रिलीज होगा? यहाँ पर in the membrane we have CF0, CF1 particle. They literally will release these protons, and while they are releasing it, they'll make ATP. तो मतलब इस स्टेप में यहाँ पर we specify protons cross the membrane and make ATP. That is what we said, photo in presence of light. Phosphorylation means making ATP. So in presence of light, we literally made ATP. Now further, cytochrome will pass electron to plastocyanin, and plastocyanin will pass this to photosystem 1. Okay, guys, if you carefully observe, electrons यहाँ से एकदम excite हुए थे, full of energy. They were literally being passed from one protein to another protein, like how you play passing pillow game in your plant cell. They are playing passing electron game between proteins. ठीक है? तो proteins के बीच में passing pillow जैसा passing electron game खेला जा रहा है, during which one of the person in that passing pillow game will keep pillow for some time with himself. सबकी energy excite हो जाती है, ये out होने वाला है, suddenly he'll pass. That is what is happening with cytochrome. It will literally create energy. It will create zeal. And the electronic energy slowly has come down. जब यहाँ पर डाउन हो गई एनर्जी, तो अगेन देयर इज अनदर लाइट हार्वेस्टिंग कॉम्प्लेक्स who will receive light radiation of 700 nanometer. They will again excite it. यहाँ पर primary electron acceptor will receive these two electrons, pass it to ultimate electron acceptor, that is NADP reductase. Guys, यहाँ पर है NADP reductase. Position के बारे में again there could be a question. What is the position of NADP reductase? NADP reductase. NADP reductase is present towards outer wall of outer membrane. Photolysis of water complex, inner wall of inner membrane. NADP reductase, outer wall of outer membrane. What is the property over here that we need to know? It is specified in your textbook that this is ultimate electron acceptor. What do you mean by this? Ultimate. आखिर में, ठीक है? At last, किसके पास जाना है? That way, ठीक है? तो electrons को किसके पास जाना है? At last, NADP reductase. Who receives it? NADP reductase is the one who is going to receive electron. Once NADP receives electrons, it will pull these two protons which came out, and this becomes NADPH2. ठीक है? That completes light reaction. अभी थोड़े नीट के क्वेश्चंस को एक्सप्लोर करते हैं. Which are the energy-rich molecules made in non-cyclic or cyclic photophosphorylation? One is ATP, second one is NADPH2. This is like two different topics in your textbook. इसको बुलाते हैं chemiosmosis hypothesis, इसको बुलाते हैं non-cyclic photophosphorylation और Z scheme. दोनों एक्चुअली एक ही हैं, बट आपको अलग अलग जगह दिखाया गया. That is why I have put both together so that you understand it. तो अल्टीमेट मकसद क्या है? मेकिंग ATP and NADPH2, and we literally dependent on light, right? तो उस वजह से इसको बुलाते हैं light reaction. Next neat point is, what is the light radiation absorbed by photosystem 2? It is 680 nanometer. And light radiation absorbed by PS1, 700 nanometer. Next neat point बताओ. In factors affecting photosynthesis, you talk about light's importance. An initial part of the chapter, Mr. Engelman will use prism, split the light into Vibgyor colors, and then he says that red light and blue light is important for photosynthesis. ठीक है? यहाँ पर two lights are important, red light and blue light. Which is more important in this? If we see this, 680, 700 comes under red wavelength. ठीक है? मतलब red light performs more photosynthesis. This is also a neat point. Followed by this, splitting of water is the one which is making oxygen, right? They can give you mathematical based question. यहाँ पर I have taken one water molecule. They'll give you 4 water molecules and ask you how much oxygen is released. Guys, can you calculate it? One water molecule, half oxygen. So 4 water molecules will produce how many oxygens? Fast calculate करके बोलिए. Cross multiply. 1 water, half O2. 4 waters. If you're not getting answer, let me know.
Two.
Exactly. 2 oxygen molecules will be released. Guys, if we say how many protons are released by 4 water molecules, क्या रहेगा आंसर? 1 water, 2 protons. So 4 waters into 4. Fast. What is the answer?
8.
8 protons. Exactly. Did you get this? तो यहाँ पर mathematical based question बन सकते हैं. Next neat question is, whose electrons were transferred in the ETS cycle or electron transport system? It was of water, not of chlorophyll A. Then next point is, why do we call it as non-cyclic? It's like your friend taking money from you. Dude, give me 100 rupees, I'll give you tomorrow. That tomorrow never comes. Right? कयामत आ जाएगी, but ये tomorrow नहीं आएगा. आपके पैसे वैसे ही गए. तो यहाँ पर वही होने वाला है. Electrons which are released from photosystem 2 are never coming back to photosystem 2. वहां से गए सो गए. अल्लाह हाफिज, टाटा, बाय बाय बोल के खत्म. ठीक है? Literally, electrons never come back. That is why we say non-cyclic photophosphorylation. Why Z scheme? In the membrane it is appearing like Z. Okay. This is about light reaction. Hopefully you understood. Ask me next part what you want.
I actually need some important chapters for my NEET.
We had already given that in a video, na beta. It's that with Botany.
I didn't get your video. I already got your video on Facebook yesterday, so I joined today.
Acha, acha. So
Yeah.
there is a video where we actually gave important chapters of all the this thing. Abhi presently ke liye, if you need
For
Botany,
major questions.
see, it's like we don't have any proper blueprint, but previous year का analyze करें तो molecular basis, biotechnology के दो chapter और reproduction वाला unit. Do not miss this in 12th syllabus. Okay? And when it comes to 11th syllabus, rank 1 preference should be given for cell the unit of life, and then morphology, anatomy. First preference में इनको देंगे आप. Okay? Second preference and third preference will be like second preference में आप 11th syllabus की बात करो, you talk about that what you say cell cycle, cell division, biomolecules, and उसके साथ साथ biological classification and plant kingdom. इसको second preference में डालिएगा. ठीक है.
Yeah.
एंड देन थर्ड प्रेफरेंस में जाना है तो देन रेस्ट ओवर चैप्टर्स को हम लोग कवर करेंगे. वही 12th की बात करो, सेकंड प्रेफरेंस में ह्यूमन हेल्थ एंड डिजीजेस, माइक्रोब्स इन ह्यूमन वेलफेयर, इसको सेकंड प्रेफरेंस में रखिएगा. थर्ड प्रेफरेंस में लास्ट यूनिट आ जाएगा. ओके गाइस, C4 cycle. Why exactly is C4 cycle occurring? What is it also known as? It is also known as Hatch and Slack pathway. Why? Because Hatch and Slack are the scientists who discovered it. Point number one. Point number two. Why is it known as C4 cycle? Cycle में बताता हूँ. Where is this occurring? It's actually occurring in the monocot plants. Where in monocot plants there are two cells which are actually involved in this. One is mesophyll cell. Second one is bundle sheath cell. Neat question is where in mesophyll cell and where in bundle sheath cell? It is actually occurring in chloroplast. Chloroplast of bundle sheath cell, chloroplast of mesophyll cell. Where in chloroplast? In the stroma. So, in monocots, in C4 cycle, we have two different cells involved and two different chloroplasts with two different stroma. तो यहाँ पर क्या होने वाला है? There are two cells. See, this structure what we have drawn is not exactly the way it is there in the so-called plant. This is mesophyll cell. And this one is bundle sheath cell. Okay. Mesophyll cell is there on the upper surface and bundle sheath cell is quite deep inside. दोनों अलग-अलग जगह है. But still, to make you understand the cycle, we show sandwich model. This model where bundle sheath cell and mesophyll cell is shown together, we call it as crans anatomy. Point to be noted that sir, these cells are not attached as shown in diagram. Mesophyll cell कहीं और है and bundle sheath cell कहीं और है. ठीक है? But to make you understand the cycle, we are keeping them together. So crans anatomy is working of mesophyll cell and bundle sheath cell together. What's going to happen over here? In the mesophyll cell, where in mesophyll cell? Chloroplast. Where in chloroplast? In the stroma. In the stroma, you have one molecule called as PEP. Full form is phosphoenol pyruvate. This contains 3 carbon atom molecule. इसी से बना word Pepsi. ठीक है? Phosphoenol pyruvate, which is there inside the cell. Which cell? Mesophyll cell. Where in mesophyll cell? Stroma of the chloroplast. यहाँ पे there is one enzyme called as PEP case. PEP case या PEP carboxylase. This PEP case या PEP carboxylase will fix carbon dioxide. Atmospheric carbon dioxide is there. It is fixing that carbon dioxide and giving it to so-called PEP. If PEP receives the carbon, it already has 3 carbons. One more carbon is attached. So how many carbon atom molecule will be made? 4 carbon atom molecule. And the name of molecule now is known as oxaloacetic acid. Oxaloacetic acid. What are the neat questions over here? Who is primary carbon dioxide acceptor? Carbon dioxide कौन accept कर रहा है? Phosphoenol pyruvate, primary carbon dioxide acceptor. Which is the first stable compound found in the cycle? Oxaloacetic acid. How many carbon atoms are there? 4 carbon atoms. Since the first stable compound formed in entire cycle has 4 carbon atoms, cycle का नाम बना C4 cycle. ठीक है? Which is the molecule oxaloacetic acid. Now, oxaloacetic acid is like mashallah type of organism. एकदम heavyweight champion है, एकदम चौड़ा, बाबा चौड़ेश्वर. ठीक है? It is very big. This molecule cannot pass through the door. उतना ज्यादा चौड़ा है. ठीक है? उतना बड़ा heavyweight champion है. ठीक है? So since it cannot pass through the door and reach to the other cell, it should go to gym, and then it will go for diet and convert itself into malic acid or aspartic acid. Malic acid or aspartic acid. This malic acid or aspartic acid, which is actually being made over here, literally is a molecule who is slim, trim, fit. It can easily pass through the membranes and come to another cell. So malic acid has come where now? Bundle sheath cell. Well, how many carbon atoms are there with malic acid? 4. The carbon dioxide which was being accepted over there, it will come and release itself in bundle sheath cell. यहाँ पे in bundle sheath cell we have rubisco. The same enzyme which is there in C3 cycle अगर आपने पढ़ा है, ठीक है? What will rubisco do? It will undergo C3 cycle, and the end product of C3 cycle is glucose. Like guys, just now in the previous cycle I told, water is donor of oxygen, carbon dioxide is donor of glucose. Hence proved. Van Neal का experiment हमने prove कर दिया. What's happening over here? Malic acid gave carbon dioxide, it gets converted into 3 carbon atom molecule called as pyruvate. This pyruvate again will go back to the mesophyll cell and convert itself into phosphoenol pyruvate. आपके टेक्स्ट में they have just specified C3 acid, C3 acid, C4 acid, C4 acid, C4 acid, C3 acid, carbon dioxide, glucose, बस. But कैसे अकर हो रहा है? I have actually shown you which is that C3 acid and where what is happening. Next neat point to this cycle is totally 30 ATPs will be utilized and 12 NADPH2 molecule will be utilized for making one glucose molecule. एक ग्लूकोज बनाने के लिए in C4, C3 में 18 ATP, 12 NADPH2, C4 में 30 ATP, 12 NADPH2. NADPH2 is common 12. C3 में 18 ATP, C4 में 30 ATP. Please remember this. नीट में पूछ सकते हैं ये वाला क्वेश्चन. नेक्स्ट क्वेश्चन क्या है? One glucose molecule is requiring how many ATP? 30. तो मैथमेटिकल क्वेश्चन बनता है 3 ग्लूकोज के लिए कितने ATP? 90 ATP. ठीक है? और if they give you ATP molecules, then till 120 ATP molecules are utilized. How many glucose molecules can be generated from this? ठीक है? This is how the questions will come from C4 cycle. Hopefully you understood. Next question पूछिएगा please. Next topic.
Sir, how many NADPH2 are used in C3 cycles?
12. It's same better. NADPH2 is common in both C3 and C4. 12 NADPH2. Okay? In both the cases it is 12.
Hello, sir.
Yeah, better. Please. Yes, Reshma. Please.
Can you please explain any important and topic about cell, the unit of life?
Cell the unit of life. पूरा chapter important है बेटा. You tell me which one. I'll write the topics side wise. See, the chapter starts by talking about cell theory. वो तो 2 statement है उसमें खत्म हो जाएगा. Then they talk about prokaryotic cell character. Then they talk about eukaryotic cell character. Which one you want? Shall I take prokaryote or eukaryote? Eukaryote बहुत ज़्यादा है शायद. Prokaryote will do. Eukaryote में अगर कोई specific cell organelle चाहिए तो बताइएगा. I'll explain that. Okay. In short, I'll try to cover entire prokaryotic part. I'll give you diagrammatic representation so that it.
Sir, I think I'm done with prokaryote and eukaryote, both of them.
Yeah, both of them are there.
Yes, sir. Any other topic, please?
Sorry?
Any other topic, please? I'm done with both of them.
And then the chapter itself is over, right? It starts with prokaryote, then eukaryotic character.
Biomolecules.
Biomolecules भी है. Which topic in biomolecules? Carbohydrates, या proteins, या nucleic acids. न्यूक्लिक एसिड्स में फॉर्मूला वो जो स्ट्रक्चर्स है ना बच्चे यूजुअली उसमें सफर करते हैं. टॉक अबाउट दैट वन. सिंपल ट्रिक देता हूं कि ऐसा क्वेश्चन भी आया तो फॉर्मूला देखकर आप लोग आराम से आंसर कर सको. सी व्हाट आर न्यूक्लिक एसिड्स. बाय द वे व्हाट इज न्यूक्लियोटाइड फर्स्ट. Nucleotide means what? Nucleotide is combination of nucleotide plus phosphate. इसमें सिंपल ट्रिक है अगेन. सी न्यूक्लियोसाइड इज व्हाट? शुगर प्लस नाइट्रोजन बेस. मतलब sugar plus nitrogen base is common both in nucleoside and nucleotide. Who is making difference? Phosphate, right? Phosphate को उठा के टाई कर दो न्यूक्लियोटाइड. T for tide, T for type. Phosphate को टाई कर दिया तो न्यूक्लियोटाइड. साइड में उठा के फेंक दिया न्यूक्लियोसाइड. If I discard phosphate, it is nucleoside. If I add phosphate, it is nucleotide. This is a simple trick. Which are the different type of nucleotide? What are the nitrogen bases? We have 4 type of nitrogen bases. Adi 9, Guva 9, cytosine. Then we have thymine. Followed by that we have uracil. Okay. These are the 4 main type of nucleotides present in DNA. And if I remove thymine and add this one, these are the 4 main type of nucleotide in RNA. Both are genetic material. Virus में RNA is genetic material. In eukaryotes, DNA is genetic material. ये भी important point है. RNA is not genetic material in cells. It is in acellular individual like viruses, viroids. अगर cells की बात करें तो it is always DNA which is genetic material. Proved and confirmed. Proof discovery हुआ था ऑसवाल्ड लावेरी से. Confirm हुआ था हर्शी एंड चेज से. याद रखना प्लीज. अच्छा, now let us try to explore this adenine and guanine. What are these two? These two are known as purines, and these 3 are known as pyrimidines. हिंदी में there is one very famous कहावत, नाम बड़े दर्शन छोटे. इसका नाम बड़ा है, दर्शन appearance is small. जिसका नाम छोटा है, it's literally a large molecule. Okay. With respect to this, if I have to talk, यहाँ पर adenine की बात करेंगे. I said छोटा नाम है but दर्शन बड़े हैं. It is bicyclic compound. 2 रिंग है इसके पास. One is the benzene ring, second one is pyrrole ring. Guanine is also purine, right? तो 2 रिंग है इसके पास. Benzene ring and pyrrole ring. Guys, you don't have to memorize entire structure. यहाँ पर कौन सा एटम है, यहाँ पर कौन सा एटम है, यहाँ पर कौन ना. 2 रिंग है, टॉप मॉलिक्यूल इज अमाइन, दैट इज adenine. Simple way. 2 rings, top molecule is amine, adenine. 2 rings, top molecule is ketone, guanine. Okay. अगर साइटोसिन की बात करो, सिंगल रिंग है, top molecule is amine, that is cytosine. How about this one? Single ring, top molecule is ketone. Even here single ring, top molecule is ketone. अरे, if both of them contain ketone, then फर्क क्या हुआ? Let us check the other name for thymine. Thymine का दूसरा नाम है 5-methyl uracil. मतलब कार्बन नंबर 5 के पास मिथाइल ग्रुप है. If this methyl group is present on carbon number 5 of the so-called thymine, uracil, and that time it is known as thymine. First point. Ketone ऊपर है, carbon number 5 पे methyl. अगर there is a side branch, thymine. Side branch नहीं है, uracil. Simple trick to identify it. कैसा भी क्वेश्चन आने दो, आराम से now you people can answer. Next is adenine always will pair with thymine, and guanine will always pair with cytosine. Molecular basis में आपने पढ़ा है between these two it is triple bond and between these two it is double bond. Next point is thymine is the reason why DNA is more stable. Stability इसकी बढ़ जाती है, whereas uracil की वजह से stability थोड़ा कम हो जाती है. highly reactive molecule होता है. फिर से next point, यहाँ पर you talk about the so-called they ask you need common pyrimidine in both DNA and RNA. Which is common one? Cytosine is common for both DNA and RNA. Exclusive pyrimidine only in DNA, thymine. Exclusive pyrimidine only in RNA, uracil. This is about the so-called that character. देन वहां पे बच्चे अक्सरियत में इन दैट चैप्टर दे गेट कंफ्यूज अबाउट प्राइमरी मेटाबोलाइट एंड सेकेंडरी मेटाबोलाइट. Okay, what is primary metabolite and what is secondary metabolite. ठीक है, इन दो के बीच में अक्सर बच्चे कंफ्यूज होते हैं बिकॉज ऑफ द एग्जांपल्स बीइंग गिवन इन नीट एग्जाम. ठीक है, पहली बात समझो व्हाट इज मेटाबोलाइट. मेटाबॉलिज्म इज सम टोटल ऑफ केमिकल रिएक्शन इन अ सेल. सारे के सारे सम एसयूएम की बात कर रहे हैं, मैथमेटिक्स वाला. सम मींस एडिशन. You have so many different type of process occurring in the cell like respiration, digestion, excretion, right? तो वो जो शन वाले वर्ड हैं, all those shans come under metabolism. दो टाइप के मेटाबॉलिज्म है, एक अनाबॉलिज्म, एक कैटाबॉलिज्म. कैटा काट्या, लार्जर पीस को काटते हैं ना, तो लार्जर मॉलिक्यूल व्हेन दे आर ब्रोकन इनटू सिंपल मॉलिक्यूल, दिस इज नोन एज कैटाबॉलिज्म. वही अगर if I assemble many smaller units together and construct a large wall, this is anabolism. छोटे मॉलिक्यूल को जमा कर बड़ा बनाओ, anabolism. बड़े मॉलिक्यूल को तोड़कर छोटा बनाओ, कैटाबॉलिज्म. बट एंड में जो प्रोडक्ट बनता है आफ्टर दिस प्रोसेस ऑफ कैटाबॉलिज्म और एनाबॉलिज्म, दे आर क्लासिफाइड इनटू टू कैटेगरीज. वन इज प्राइमरी मेटाबोलाइट, सेकंड इज सेकेंडरी मेटाबोलाइट. प्राइमरी मेटाबोलाइट किसे बुलाते हो आप? Any such chemical which is prepared by the plant and used by plant itself. या फिर कोई एनिमल कोई केमिकल बना रहा है अपने बॉडी में and it is utilizing itself for its own purpose. For example, glucose, starch. This is a chemical prepared in the plant and used by plant itself. Primary metabolite. We talk about secondary metabolites, drugs that we take. Human health and diseases में आपने पढ़ा opioids, cannabinoids, nicotine, right? कोका. ये सारे जो केमिकल्स हैं, हशीश, मरुआना, गांजा, ये सारे केमिकल्स आर यूज्ड एज मेडिसिन. ठीक है, तो प्लांट इज प्रिपेयरिंग दीज केमिकल्स, बट प्लांट खुद नशा नहीं कर रहा है, दूसरे लोग नशा कर रहे हैं. इट इज फॉर अदर्स. परफ्यूम इन द प्लांट्स. डू यू थिंक प्लांट अपने आप को रोजाना जो है डीओ लगाकर परफ्यूम लगाकर घूमने जा रहा है? ना, दैट इज फॉर अदर ऑर्गेनिज्म. प्लांट इज प्रिपेयरिंग इट, बट इट इज फॉर अदर्स. Oils in the plants, sunflower oil, groundnut oil, all the oils that we are taking, castor oil. The oil in the plant is produced and it is not using by itself. It is for other organism. इसको बुलाते हैं आप secondary metabolites. ठीक है. Next question पूछो बेटा. Next topic.
सर आप प्लीज ट्रांसक्रिप्शन एक्सप्लेन करेंगे?
Yeah, sure. So what is transcription? Phenomena where DNA will decode itself into messenger RNA. Messenger RNA is a script, message carry करने वाला RNA. See, whenever we are making any message and this message has secrecy, you can make it in a transcript. मतलब जो ऐसी लैंग्वेज जिसे डिकोड करना पड़े, आम लोगों को इसके बारे में नॉलेज ना हो. So this phenomena where your genetic material is making a transcript, top secret letter बना रहा है. ठीक है, this top secret letter what they are making, एक ऐसा है like writing a love letter. ठीक है, and love letter को जो आपने लिखा है, it is in coded information, coded language में लिखा है love letter. And this love letter what you have made, you threw it in the home of your partner. ठीक है, उनके अब्बू के हाथ को लग गया ये लेटर. अब्बू जब पढ़ेंगे लेटर, since you have written in transcript, he is not at all able to identify it. Top secret वाला लेटर. ठीक है, and then only if you get that letter, then आपको पता चलेगा उसमें लिखा है जान मेरी जाने मन बचपन का प्यार मेरा भूल नहीं जाना रे. Right? Because you know how to decode it. So we are making a transcript. Top secret letter बना रहे हैं inside a cell to kill viruses, to make a protein that is known as transcription. On our DNA, this is double-stranded DNA molecule. Okay, here we have 3 prime, 5 prime, 5 prime, 3 prime. Towards 3 prime end of template strand, we have a special sequence. अपने DNA पर, आपके बॉडी में, मेरे बॉडी में, बैक्टीरिया की बॉडी में, हर ऑर्गेनिज्म में ये प्रोसेस अकर हो रहा है to make sure that we fight against viruses and develop some good proteins. This sequence over here is known as promoter. ठीक है, the one who promotes the activity. On the other side, you have terminator. the one who stops. And in center we have structural gene. Okay. This is known as template strand and this is known as coding strand. सबसे पहला नीड क्वेश्चन इज अबाउट द स्ट्रक्चर इटसेल्फ. The last two, promoter sequence is present towards what end? किस साइड है? It is towards 3' end of template strand. Promoter sequence is towards 3' end of template strand. Whereas terminator 5' end of template strand. या फिर coding strand का पूछो 5' end of the coding strand is your promoter, 3' end is terminator. First need question. Second question is what is work of promoter, terminator? See, we were talking about love letter, right? यहाँ पर the person who promotes, तुम्हारा एक फ्रेंड है जो बोलता है dude try कर, so okay dude चलना try कर. The one who is pushing you towards what is writing letter, he is promoter. एक और मौली साहब वाले फ्रेंड है या फिर एक सेम टाइप ऑफ फ्रेंड है, he is like why are you doing this, don't do this dude, stop it. Terminator. The one who stops is terminator, the one who promotes is promoter. So two type of sequences, and the central gene is the actual gene which is gonna make me mRNA. This is about the structure. अब बात करते हैं about transcription. How does it occur? It occurs in 3 steps. First step is initiation. Name itself is telling you it starts the process. How is it starting? Guys, यहाँ पे at the promoter sequence we'll have RNA polymerase attaching to itself. RNA polymerase. There are 3 types of RNA polymerase. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. 3 टाइप के RNA polymerase हैं. अगर RNA polymerase 1 will go and bind over here, that will make rRNA. अगर RNA polymerase 2 will go and bind here, it makes mRNA. अगर RNA polymerase 3 will go and bind, it makes tRNA. Right now I want to make what? mRNA, right? So I'll take RNA polymerase 2, which will come and attach over here. जैसा RNA polymerase 2 will go and attach over here, it creates transcription bubble. एक छोटा सा bubble बनता है, which is known as transcription bubble. Once transcription bubble is made, now sigma factor will bind to it. I would like to relate this to biomolecules. Biomolecules का last page आप चले जाओ, वहां पर you talk about cofactors. What are cofactors? Non-protein part which will associate with protein and activate it. Okay, this is known as so-called cofactors, non-protein parts. Sigma factor is such kind of cofactor who is activating whom? An enzyme known as RNA polymerase. जैसा sigma factor binds to this, RNA polymerase now starts reading template strand. ठीक है, आप अगर टेम्पलेट्स एक रैंडम सीक्वेंस लिखते हैं यहाँ पे, से A T G C T A G C T. This is a random sequence. 3' is here, 5' is here. This is template strand. नीट में ऐसा क्वेश्चन देंगे आपको, template strand, and then ask RNA polymerase क्या बनाएगा, ट्रांसक्रिप्शन में क्या बनेगा यहाँ पर? A will pair with whom? U. Guys, remember this. A will pair with U. T will pair with A. G will pair with C. C will pair with G, T will pair with A, U, T, G and A. सीक्वेंस भी हमें इंपॉर्टेंट है. आप सब ये बड़ी गलती करते हैं. देखिए बेटा, टेम्पलेट स्ट्रैंड दिया गया है, they are asking you to write about the sequence of so-called RNA molecule. Instead of A, you are writing U. Agreed. लेकिन पोलैरिटी भी चेक करनी पड़ेगी. अगर यहाँ पर 5' एंड है एंड 3' एंड है, ओनली देन दिस इज राइट आंसर. अगर यहाँ पर 3' एंड है एंड 5' एंड है, दिस इज रॉन्ग आंसर. तो पोलैरिटी शुड आल्सो बी चेक 5', 3'. दे विल गिव यू मिरर इमेज ऑफ दिस आल्सो. ठीक है, अगर गलत ऑप्शन दें तो मिरर इमेज देंगे, वो आंसर रहेगा. दिस इज अबाउट सो कॉल्ड एडिशन. So RNA polymerase literally reads template strand and starts adding nucleotide and makes a long chain of mRNA. जैसा long chain बन गया, this RNA polymerase now reaches where? To terminator sequence. जैसा terminator sequence को it will reach, rho factor will bind it. Initiation में this was the activity which was happening, right? Elongation में We made a long chain of nucleotides. Now it's time for termination. What happens in termination? RNA polymerase reaches terminator sequence. यहाँ पर rho factor will bind it. Once rho factor binds to this will stop the process of transcription. First thing that this will do is release mRNA. जो RNA molecule यहाँ से बना है DNA से उसको रिलीज कर देंगे. Once it has been released, this ends transcription. Hopefully you understood. एक दो नीड पॉइंट बोलता हूं. देखिए यहाँ पर I said template strand का sequence देके they'll ask you about mRNA. They can also give you coding strand and ask about mRNA sequence. अगर coding strand दिया गया, the only difference between coding strand and mRNA is instead of U you have T. बाकी सब same. सब सेम रहेगा. पोलैरिटी भी सेम रहेगी. 5 प्राइम 3 प्राइम. यहां पर पोलैरिटी मत चेंज करो. पोलैरिटी सेम, सिर्फ U एंड T के बीच में डिफरेंस आएगा. बाकी का सब सेम रहेगा. इफ दे गिव कोडिंग स्ट्रैंड, इफ दे गिव टेम्पलेट स्ट्रैंड, एक तो कॉम्प्लीमेंट्री बेस बनाना है, दूसरा पोलैरिटी भी चेंज करना है. ठीक है. We should stop here. We'll have what is the next session soon. Inshallah, we'll keep you updated regarding that. Okay. Meanwhile, do you have any doubts in the topic what we covered today? Any objective question that you have come across while you are just going through and you did not understand that? Please let me know. I'll clarify it.